Welcome to our presentation on the Cre Lox System, a revolutionary tool in genetic engineering. This technology enables precise DNA modifications with unprecedented control. We'll explore how this elegant molecular system works and why it has become essential for modern biological research. The Cre Lox system was identified in bacteriophage P1, a virus that infects bacteria. It was first described in 1981 by scientist Nat Sternberg. Initially, its purpose was to maintain viral DNA during bacterial infection. The system consists of two essential components that work together: Cre recombinase enzyme and Lox P recognition sites. Together, they create a precise genetic editing tool with remarkable specificity. Cre recombinase recognizes and catalyzes recombination between specific DNA sequences called Lox P sites. Lox P sites have a unique molecular architecture, including inverted repeats, a spacer region, and directional function. The inverted repeats are two 13BP sequences facing opposite directions, while the spacer region is an 8BP asymmetric sequence between repeats. The asymmetry determines the recombination outcome. Cre recombinase is a 38 kDa enzyme with 4 subunits that form a tetramer around DNA. It belongs to the tyrosine recombinase family, using tyrosine residue in catalysis. The enzyme recognizes Lox P sites, cleaves DNA, and rejoins strands in new arrangements. Cre Lox recombination can result in 3 types of events: inversion, deletion, and translocation. Inversion occurs when Lox P sites face opposite directions, flipping the DNA between them. Deletion happens when Lox P sites are in the same orientation, removing the DNA between them. Translocation occurs when Lox P sites are on different chromosomes, exchanging segments. The timeline of genetic engineering evolution begins in 1981 with the initial discovery in bacteriophage P1 by Nat Sternberg. In the early 1990s, the first applications in mammalian cells and transgenic mice were developed. By the mid-1990s, conditional knockout technology in mice emerged. From the 2000s to the present, applications have diversified to various organisms and fields. Conditional mutagenesis operates on 3 principles: spatial control, temporal control, and inducible control. Spatial control restricts gene modification to specific tissues. Temporal control activates gene changes at specific developmental stages. Inducible control triggers modifications using external compounds. Designing a Cre Lox experiment involves several steps. Flox your gene by creating a mouse with Lox P sites flanking your target gene. Introduce Cre by crossing with Cre-expressing mice or delivering Cre via viral vectors. Validate recombination to confirm DNA modification occurred as expected. And analyze phenotype to study the effects of your genetic modification. Cre transgenic mouse lines include global expression lines where CMV Cre or ELA Cre express recombinase in all tissues, neural tissue lines like Nestin Cre targeting neural stem cells and derivatives, organ-specific lines such as albumin Cre targeting hepatocytes in the liver, and cardiac lines like alpha MHC Cre targeting cardiomyocytes in the heart. Inducible Cre systems include the Cre system, which fuses Cre with estrogen receptor for tamoxifen activation, and Cre2, an enhanced version with improved nuclear translocation efficiency. Temporal control is achieved by administering tamoxifen only when recombination is desired. Tissue-specific and developmental control involves using common promoters for specific applications. For example, MY6 is used for cardiac development and disease, PDX1 for diabetes research, CAM-KII for learning and memory, and K14 for wound healing and cancer. Viral delivery of Cre recombinase can be achieved using vectors like adeno-associated virus, AAV, lentivirus, and adenovirus. Each vector has unique properties for specific applications. Delivery methods include direct tissue injection, intravenous delivery, and ex vivo cell transduction, depending on target tissue accessibility. Dual recombinase systems involve two recombination events, the Cre Lox system for the first event and the FLP-FRT system for the secondary recombination event. The combined result is precise genetic modification at the intersection of these events. Applications in mouse genetics include knockout models to delete specific genes and study their function, knock-in models to insert new genes or modified versions, reporter models to add fluorescent proteins for cell activity tracking, and developmental studies to study gene function during specific growth stages. Lineage tracing and cell fate mapping involve multicolor labeling using rainbow technology to create unique cellular color codes, developmental tracking to follow cells from progenitors to mature states, and single-cell resolution to visualize individual cell morphology and connections. Disease modeling with Cre Lox includes cancer models to delete tumor suppressors, activate oncogenes, and study cancer progression. Neurodegenerative disease models focus on conditions like Alzheimer's and Parkinson's. Metabolic disorder models include diabetes, obesity, and liver disease studies. Timing is crucial in Cre systems. Constitutive Cre is active from the moment of expression, which can cause embryonic lethality and developmental compensation. Inducible Cre is activated only when needed, allowing controlled timing, avoiding developmental issues, and studying gene function in adult tissues. Tools for reporter and conditional alleles include the ROSA26 locus, which is ubiquitously expressed as a genomic safe harbor, the I9 reporter for strong TD tomato expression after recombination, and rainbow constructs for multiple color options in cell labeling. Leaky Cre expression presents challenges such as unintended recombination in many Cre lines, with a typical leakage rate of 5–15%. Germline risk is 100% when recombination occurs in germline cells. Mosaic expression results in patchy recombination patterns in target tissues, with a rate of 30%. To evaluate recombination efficiency, we utilize several methods. Genomic PCR helps detect deletion events by targeting primers flanking LOXP sites. Reporter visualization uses fluorescent proteins to indicate successful recombination. Quantitative PCR measures recombination percentage in tissue samples, and protein analysis through Western blot confirms loss of protein expression. Understanding off-target effects and toxicity is crucial in genetic experiments. This pie chart represents the distribution of various off-target effects and their associated toxicity levels, highlighting the importance of precise targeting and minimizing unintended consequences. Implementing best practices in experimental design ensures reliable results. Essential controls include Cre-only and Floxed-only animals, as well as wild-type animals. Breeding strategies should maintain Cre transgene paternally and check for germline recombination, using heterozygous Cre when possible. Validation methods encompass multiple recombination assays, reporter line test crosses, and careful phenotype analysis. When troubleshooting common problems, it's important to address issues such as low recombination, unexpected phenotypes, and lack of phenotype. For low recombination, check Cre expression levels and patterns. Investigate off-target effects or genetic background for unexpected phenotypes. Validate targeting strategy and gene redundancy if no phenotype is observed. Ethical considerations and animal welfare are paramount in research. The 3 rupees principle, replace, reduce, refine, guides animal use. Regulatory compliance involves IACUC approval and adherence to GMO guidelines. Welfare monitoring includes regular health assessments and humane endpoints to ensure ethical treatment of animals. The Cre Lox system is expanding beyond mice to other organisms. Zebrafish, with their transparent embryos, are ideal for visualizing recombination events. Drosophila serves as a powerful genetic model for developmental studies. Plants offer expanding applications in agricultural research, showcasing the versatility of Cre Lox technology. Comparing CRISPR and Cre Lox systems reveals their complementary strengths. Cre Lox offers site-specific precision at LOX-P sites and predetermined genetic rearrangements, requiring transgenic lines for preparation. CRISPR, Cas9 provides programmable targeting with various edits possible and direct delivery options, suitable for gene editing and corrections. Recent advancements in recombination technology include split Cre, optogenetic Cre, synthetic promoters, and chemical genetic systems. Split Cre technology activates Cre fragments only when co-expressed in the same cell. Optogenetic Cre allows light-activated recombination for millisecond precision. Synthetic promoters are engineered for precise expression patterns, and chemical genetic systems enable designer drug-controlled recombination with minimal toxicity. Future directions in recombination technologies are promising. Single-cell precision targets individual cells within complex tissues. Therapeutic applications adapt recombination for human gene therapy and disease treatment. Multiplexed editing modifies multiple genes in controlled patterns, and AI-guided design uses machine learning to optimize recombination strategies. In summary, the Cre Lox system remains a cornerstone of functional genomics. Its evolving methodology enhances precision and capabilities. Versatile applications range from basic research to disease modeling and potential therapies. Future integration with new technologies will expand research frontiers, combining strengths for greater advancements.